Picking up on anabolism and catabolism. So these two things, as you might imagine, are interrelated. So as we look at this, so catabolism again is going to be things that are breaking other things down. So this is going to be oxidative, it's going to be exergonic, it's going to give us energy, it's going to take sugars and proteins and fats and break those down into a variety of different things. So these are going to be energy poor end products. So while there are a variety of different products you can get from catabolism, realize that those are going to be degraded all the way down eventually into water, carbon dioxide, and ammonia. When all of that gets degraded, you're going to get a lot of chemical energy out of this. You're going to get NADPH as a reducing power, and you're also going to get ATP for energy. And then from there, we're going to use those pieces to basically provide the energy we need for anabolism to go ahead and make things. Because the body's going to need to make proteins, it's going to need to make its own fats, it's going to need to store a whole bunch of different things. So in order to make all of those macromolecules, it's going to need a different power to do that. So it's going to take all of those bits of energy and various pieces from catabolism and utilize those in making a variety of things in metabolism. So a variety of different precursor molecules to make those larger macromolecules. As we look at these two things, they're not mutually exclusive. So that's why we kind of took a look at that big biochemical map, is that there's a lot of different ways these things can interconnect together. So even though the catabolic pathways converge to those few end products, and your anabolic pathways diverge to make a whole bunch of different things, some pathways do serve in both of them. So again, just think about that. Your catabolic pathways are going to converge into making those few end products. Your anabolic pathways are going to diverge into a variety of different things. But again, some pathways are going to serve both. Even though they get some of the same products, they're not necessarily the same pathway, and some steps are common to both. We're going to look at that especially as we look at glycolysis, which is the breakdown of glucose, and gluconeogenesis, which is the making of glucose. We're going to look at both of those pathways, and what you'll notice then, and that does happen relatively often, is that some of those steps are identical. Not just, really similar, but will actually use the same enzymes, use the same pieces, and will actually be identical. But some of those will also be different. This is to ensure that each pathway can actually be favorable. Because remember, you are still in a cell, and you need some way to actually drive these reactions to go one way or the other. So not only is this going to help allow these different pathways, either breaking things down or building things up, to be favorable, but this is also going to allow the cell to allow regulatory mechanisms so that you can turn one off or the other. And again, this is talking about things in a really broad way so that we can basically lay the groundwork and foundation to talk about things in a more specific way. So again, we're going to converge to very, very few products. So as you look at all of these wonderful things, on the very top box, you're taking proteins, polysaccharides, and lipids and breaking those down. So as you break those down to component building blocks, your proteins go to amino acids, your polysaccharides are going to wind up going to glucose, your lipids are going to go to glycerol and fatty acids. So from those three things, they're all going to actually wind up going to mostly to acetyl-CoA or pyruvate. Different parts, depending on how the amino acids are broken down, those may very well enter the citric acid cycle. And then with all of those amino acids, that amino group is going to give you that ammonia. So you're going to basically wind up with ammonia from your citric acid cycle, which we'll also call the TCA or tricarboxylic acid cycle. You'll also be going to water and carbon dioxide. So you've got basically anything that you take in, you're going to get down into only 3 different products as those go. So you take in a wide variety of different things, and as far as catabolism is concerned, you only get 3 different end products: ammonia, water, carbon dioxide. So again, just a reminder, they're all related. So this is kind of that good slide to kind of see and be like, oh yeah, catabolism breaks things down, but gives a whole lot of energy. Anabolism is going to then take that energy and make new things out of it. So one of the things that's here a whole lot is you see a lot of ATP. So I've already mentioned the fact that we're going to be talking about that a lot, and this is a really good time to bring back that notion of ATP. So we're going to be using ATP as basically the energy currency of the cell. You've probably heard that before. And just like currency, we expect it to change hands really often, move around a whole lot, not stay in one place. So those phototropes would actually transform light energy into chemical energy of ATP. So yes, plants still use ATP. In heterotrophs, that catabolism is going to produce ATP, which is going to drive the activity of cells. So not a surprise that ATP is again playing a major role. And can also happen when you're looking at ATP carrying energy from photosynthesis or catabolism to energy requiring processes. So we're going to have that transfer of ATP where it's being produced from catabolism or photosynthesis and then being transferred over into something like an anabolism pathway where you're creating some sort of biomolecule. So again, this pathway is just kind of trying to show you that same general notion that catabolism is going to produce things like water and carbon dioxide, but also ATP. ATP is then going to be used, is going to be hydrolyzed to do a variety of different things, whether that is motion or working on keeping gradients going, or working on actually making different molecules. That ATP, when it's used, is usually going to go back to ADP and that inorganic phosphate, but combined with fuel and water, then could basically be going back into the cycle. This could also then go through photosynthesis and back to ATP. So just kind of depending on what cycle you're in. So you guys have seen this before. So just a reminder of what this looks like. When we look at ATP in catabolic reactions, these can actually be directly coupled. So this is what we call substrate level phosphorylation. So this is where you can see on the left, there is a molecule of phosphoenol pyruvate, or PEP for right now, and it has that phosphate on it. And by using an enzyme, it's going to take ADP and take that phosphate group off of the substrate, off of PEP, and put it onto ADP to make ATP. So what's happening there is we call it substrate level phosphorylation. It's going to differentiate it from the electron transport chain and the proton motive force and an ATP synthase, which all of that's going to be on the last exam. So I understand that at this point you're like, compared to what? So we are going to see in glycolysis a lot of substrate level phosphorylation. So just realize that at this point it seems like that's the only way we would do it, but the cell has another trick up its sleeve that we're going to see later. And again, just to remind you that we've talked before about coupling thermodynamic processes. So you can see at the bottom that going from phosphoenol pyruvate to pyruvate is really well exergonic, but making ATP is endergonic. So when we couple those two things together, we still have the thermodynamically favored oomph to basically get this reaction to go forward, but making that ATP is not favorable by itself. So it's coupled in this case to releasing that phosphate off of PEP. Catabolism is oxidative and anabolism is reductive. So we are actually going to use oxidation and reduction to talk about these pathways and certain things that are happening. But I'm well aware that oftentimes redox is something that we kind of hope stays under the rock that it lived under when we were in Gen Chem 2 and never comes back again. But it's actually useful to talk about it. So this one might be a review, but it's a good review to have. So oxidation-reduction reactions are usually called redox reactions. This is going to be talking about the transfer of electrons between reactants. So technically your oxidation numbers will be changing. Sometimes you can actually see it in a charge, and sometimes it's an oxidation number. Oxidation is loss of electrons. So we talk about this as being oil. Oxidation is loss, and reduction is gain. Reduction is gain of electrons. When I think about this, I think of this as being a reduction in charge. So we usually look at something becoming more negative or becoming less positive, but reduction, it's a reduction in charge. And so redox reactions are going to occur because different elements have different tendencies to gain or lose electrons. We'll talk more about what that really means later on. But for now, we're just going to talk about it that this is a thing that happens. When we're talking about oxidation and reduction, there's different ways we can talk about this. We can either talk about something being an oxidizing agent, as a species that accepts electrons, something that gets reduced, or a reducing agent, which is something that's going to be able to donate an electron. So it's going to be able to give up that electron, or something that will be able to be oxidized. So we either have things that can take electrons or things that can give electrons. And in doing so, we can either talk about essentially what's happening to it. something that either gets reduced or gets oxidized, or something that helps the oxidation to happen. If it's helping the oxidation to happen, it's going to be reduced itself because it's going to accept those electrons, but that makes it the oxidizing agent. We'll talk more, and so the next slide's also going to help with this. So NADH can be oxidized, NAD plus can be reduced. So you'll notice that that NAD plus has a positive charge on it, so it can accept more electrons. NADH is nice and neutral. It has extra electrons that it can give up. So when we think about that, think to yourself for a minute, based on those definitions, if my NADH can be oxidized, think for yourself for a minute, which one is that going to be, the oxidizing agent or the reducing agent? What you see here is you see a particular chemical reaction. This is an old one I borrowed from Gen Chem. And do go ahead and take a minute, pause the video if you need to, and figure out what goes where. What's the species oxidized? What's reduced? What's the oxidizing agent? What's the reducing agent? Trying to get some of that terminology of what's happening. But what you should be able to see here is that zinc goes from being neutral to being positive. So because it became more positive, it's oxidized. It lost electrons. Your copper 2+ goes from being positive to being neutral. It therefore gained electrons, so it's being reduced. Therefore, we basically swap those terminologies. Copper 2+ is an oxidizing agent. It lets the oxidizing happen. It's going to accept and take electrons, and because it's taking electrons, it therefore lets the oxidation happen. It helps zinc be oxidized. On the flip side, zinc is basically providing electrons. So zinc is just going to give away its electrons, and as zinc gives its electrons away, it allows reduction to happen. So zinc provides the electrons that are needed for reduction to actually occur. So coming back to biochemistry. So that was our little foray back to Gen Chem 2. Now we're back to biochemistry. NAD+ is going to collect electrons that are released in catabolism. So again, NAD+ is going to serve as that is going to be reduced. So NAD+ is going to be reduced because it's going to go ahead and collect the electrons. It's going to take electrons. It's going to get reduced. So because of that, the substrates of catabolism tend to be good sources of chemical energy because their carbons are already reduced. So as your catabolism is happening and as everything is basically coming apart, those are going to basically, we're going to save and try to hold on to that reductive power by reducing NAD+. So as catabolism is happening, you get oxidation reactions out of that, and we're basically going to, because the parts of catabolism need to be oxidized, we're going to have NAD+ be reduced along the way. So that's going to often be done in the term of hydrides. And that's what's going to allow the NAD+ to be reduced to NADH. So NADH can then turn past those reducing equivalents to other acceptors. And at the very end of everything, we're actually going to take oxygen, which is often known as that ultimate oxidizing agent. It's the final acceptor of electrons and be reduced to water. So this is a great time to take a moment and again, see if you can figure out from this, just looking at some of the charges, what we've already talked about, if you can figure out what's being oxidized and what's being reduced. So what's happening again is that NAD+ is being reduced to NADH, which means that your lactate is being oxidized to pyruvate. So this is still a catabolic reaction where we have things being degraded little bit by little bit. And whenever you have a reduction reaction, you always have an oxidation reaction. Those two things have to go together. We're not going to have electrons just float off into space. So anytime you have a reduction reaction, you've got to have an oxidation reaction to go with it. Oh, so what about the agents? If I wanted to know where my reducing agent is and where my oxidizing agent is, where are those? So keep in mind that whenever we're talking about the agents, we're always going to be talking about the reactants, the substrates, the beginning part. Those agents are always in the beginning. So here, when we're talking about an oxidizing agent, that's going to be that NAD+ is our oxidizing agent. It's what allows the oxidation to happen. It accepts and takes those electrons. Your lactate technically is a reducing agent. your NAD plus has to get its electrons from somewhere, and they're going to come from lactate. So whatever is going to give those electrons up technically counts as that reducing agent. So the nicotinamide is oxidized when it has that positive charge and reduced there on the right in blue. The rest of the molecule does stay the same. In case you wanted to know how to pronounce this is nicotinamide adenine dinucleotide. There are often different ways to pronounce things, but that's how I'm going to go ahead and do it, is that nicotinamide adenine dinucleotide. But we're going to honestly just call it NAD+, because it's a lot easier to say. As mentioned previously, the NAD+ is going to be what collects the electrons released in catabolism. So what you'll notice is that there's a lot of different atoms hanging out in here, but the only thing that's really changing is that part that's highlighted there at the very top. So hydrogen electrons are going to be released in the course of oxidative catabolism and transferred as hydride ions to that nucleotide there at the very top to form that NADH, NAD hydrogenase reaction. And because there's going to be a variety of different catabolic reactions that are going to be oxidative in nature, those oxidative reactions are often going to be coupled to the reduction of NAD plus to NADH. So you're often going to see a variety of reactions that are breaking things down produce NADH and give us that reductive power that we'll utilize in a later thing. It's going to be the electron transfer. Just a reminder, NADPH is often going to provide the reducing power for anabolic processes. So we were just talking about oxidation and reduction and a lot of NADH. And all of those involving the fact that when you're breaking things down, you're often going to have catabolism and being the oxidative catabolism. So anabolism, when we're building things, is often going to be reductive. So therefore, we're going to need something to be that counterbalance. So just like when we had breaking things down, being oxidative, and NAD plus going to NADH to counterbalance that, when we have things that then are going the other direction, we need reductive reactions to happen. We're going to need an oxidation reaction to couple with that. It's technically usually going to be from NADPH. So this is going to be something that's going to help carry electrons from catabolic reactions to anabolic reactions. In photosynthesis, light energy is used to pull electrons from water and transfer them to NADP+. That's again one of the reasons why plants are really neat, is plants can actually split water all by themselves with the help of light, and plants then get oxygen as a byproduct. So plants really can actually break water down, and as they do that, they actually produce oxygen. Plants are cool. Here's a look at NADPH. So again, the only thing that's really changed is that bottom portion where you see the phosphate group. Since NADPH is going to provide the reducing power for anabolic processes, we again get another kind of cycle going where we can have some sort of fuel that is being broken down with catabolism into some sort of oxidized product. Since that gets oxidized, that's often then going to reduce NADP plus to NADPH. When that's reduced, then we can actually use the NADPH to be the oxidized, to get oxidized. So then when we have reductive biosynthetic pathways, you need something that's going to be oxidized to counterbalance that reduction pathway. So I highly recommend you guys take this slide and either through Skype or whatever you want, or talking to a stuffed animal, talk through the oxidation and reduction that's happening here. This slide looks really simple, but just trying to talk out loud with oxidation reduction, it's a really good practice in order to get it right.